I want to introduce Dr. Harri VanHala. He is the Deputy Director for the National Center for Earth and Space Science Education and oversaw all flight operations for SSEP, so you probably know Harri very well. There you go.
All right, thank you. All right, it's been a fantastic morning. I think the presentations have been really wonderful, and I've really enjoyed hearing the first-hand experiences of students. Some of you have just preparing your experiments for flight in the fall. Some of you have experiments in flight, and including this last one, have already results to discuss. So that's all been fantastic to hear. I wanted to spend a few moments this morning talking a little bit about how these experiments actually come to be from the moment they are selected for flight to the point where they actually are flight experiments. Because it's not just a simple process of saying this is what we want to fly and then you just put it on the shuttle. In some cases, another ferry vehicle for our ISS missions, you actually want to do something with the experiments to make sure that you get the most out of them and they also are ready to fly. So I wanted to spend a few moments about talking what it takes to get the experiments ready for flight. So briefly, I'm going to be very brief about it. A lot of you are familiar with different stages of this process, obviously because you have flown experiments or are getting ready to fly. But I wanted to explain to everybody what all is involved. So when you send in your proposals for review, and final experiment selection, there are several phases that goes through, and I'm going to be going back and forth between these two screens. So I'll briefly explain a few words about the review process and how we select an experiment, including the confirmation of a tentative selection for actual flight, some of the process that we have to go through with NASA to make sure the experiment can fly. The most important one is obviously the NASA flight safety review to make sure that the experiment is safe to fly aboard the shuttle or to the International Space Station. We then have to schedule it for flight for the crew aboard the International Space Station. And finally, of course, you have to prepare the experiments for flight. The students teams prepare the experiments for flight, send them to NanoRacks. NanoRacks prepares the payload, and the payload is then transported now to the ISS. So as you can see, I'm sort of focusing on the current state of SSEP. A lot of the processes are the same, both previously for the shuttle flights, but especially now for the International Space Station missions. And I'm going to use SSEP Mission 1 to the ISS as a sort of a template for showing how things go about. So let's talk about some of these processes. So as you've heard from some of the teams already, the basic idea of SSEP is that the communities taking part in the program hold a design competition in the community. Student teams prepare proposals within a community. The Step 1 review board selects up to 3 finalist proposals, which they send to us for review. We then convene a Step 2 review panel, which includes scientists and science educators, engineers from a variety of institutions, universities, educational institutions, research labs, from usually around the Metro DC area, sometimes from further away. For example, for the latest review, we had researchers from the NASA Jet Propulsion Laboratory in Pasadena. So we can do that nationally, but especially locally, because Step 2 review panel meets in Washington, D.C. We've had review panels meet here at the National Air and Space Museum the last couple of times, but we also meet at the Carnegie Institution of Washington, which is about a mile away from where we are now. But the idea is that we really review these proposals very thoroughly to come up with what we think is the best experiment within the constraints of the experiment. Sometimes they are very good experiments in principle, but there's some issues with the way that they have been designed that don't work well within the constraints of the mini laboratory or the scheduling about the International Space Station. But eventually we come up with an idea of the tentatively selected experiment. But that's not the end of the selection process. After something is tentatively selected, there's still a lot of work to be done. First of all, the student team that has been tentatively selected to fly has to answer any questions posed by the review team. Somebody mentioned an earlier talk about the question of wanting to run an experiment with the bacteria throughout the mission, but the review panel asked questions like, do they have enough resources? Do the bacteria have enough resources to last throughout the flight? And as a result of these questions, student teams have actually revised the experiments to include something like a fixative to stop the experiment at the specific point. So these questions can be very useful for the team, but they also can sometimes are means of making sure that the experiment selection is valid. The second issue is to finalize the list of experiment samples to make sure that the samples you want to fly are the ones you really want to fly when it comes time to prepare the experiment for flight. And finally, you also need to then confirm special handling requests and timeline aboard the ISS. And that is usually done in the form of the experiment details form, which a lot of you are familiar with, and I've included a sample here. And that actually is from mission 2, that is for an experiment that's going to fly this fall. And once you have answered all these questions, once you have specified the experiment details that we need to specify, the tentative selection can be confirmed, and then you can be congratulated for actually getting an experiment for flight. However, that's still just the beginning of process. One of the important parts is to make sure that all experiment samples are specified very precisely. And what do we mean by that? And actually this goes with one of the teams that presented their experiment design for an experiment to fly in SSEP mission 2 this fall. For example, if you want to fly Lactobacillus acidophilus and milk powder, you actually can't say it just like that. You have to say it very precisely in what you want to fly. This mixture, in short, ampule A, you have to say it's a mixture of these two substances, what form they are, powdered in this case. You have to say what ratio they are. You have to say what is the concentration of the bacteria you want to fly, in this case in terms of colony forming units. And you also have to say exactly what the precise ratio of everything is. That is important because one of the crucial tests for our experiments is to make sure that they can pass the NASA flight safety review. And NASA flight safety review is basically intended to make sure that anything that flies aboard NASA vehicles into space, aboard the shuttle, or as part of the NASA payload to the International Space Station is safe for astronauts to handle. Obviously, we don't want to send anything dangerous into space. So this is a very, very rigorous process, and that's why we need to make sure that all the concentrations, all the details for experiments are very precise. And as those of you who have come through this process already, and all of you have, all the flight experiments team have, what you need to provide for this process is the final samples list with the precise concentrations we talked about. You also need to provide test results for human samples that you may want to fly. But also one crucial part is these material safety data sheets, which basically give give information on how safe different substances are for handling. And all of the teams who participated in Mission 2 actually were very good in finding the material safety data sheets for your experiments. So thank you, everybody, for doing that. That really was very helpful. We are now, the good news is that to date, all SSEP samples that have been submitted to the NASA Flight Safety Review have passed it. And we really had to have NanoRacks and their expertise to thank for that, because they have been very good in making sure that everything goes through the NASA Flight Safety Review. The samples for Mission 2 are currently in that review process, and we should hear in the next 4 to 6 weeks how everything goes with Mission 2. But so far, everything has been very successful, and it's in large part to the precise way that you have been able to provide your experiment details, and also the great help from NanoRacks. The second part of the experiment preparation that is important at this point is to make sure that we come up with a timeline of activities aboard the ISS. This wasn't a big deal for the space shuttle's flights, but it is very big for the ISS, where we actually have to have the crew handle the samples by handling the mix sticks or fluids mixing enclosures, depending what you want to call them, and activating and deactivating experiments. And a couple of things that we do with that process, and this is really driven by NanoRacks and their liaison to the ISS scheduling office. And I just show you a couple of diagrams here that we prepare at this point. And one that shows the different experiments and at what point from the beginning of the mission. This basically is a timeline, general timeline for the payload aboard the station. So this is actually SSEP Mission 1, the ISS. At one point when we were supposed to be at the ISS, you know, it was delayed a few times. The launch was, we started a little bit later than in this particular schematic. But basically from the beginning to the end, we plot each experiment, when it's supposed to start, when it's supposed to end. In this case, as you can see, a lot of experiments are started at the beginning of the mission and then stopped. And some of the experiments are started at, you know, at some point before landing, and then they probably remain active all the way until landing and coming back to Earth. So that is one task that we do with the ISS scheduling office. The other thing is to make sure that we label, provide a schematic for the astronauts to know which mix sticks or FMEs to handle at which point, and which part to bend at which point. Somebody asked earlier today, how do the astronauts know which ampule, short ampule A and B to crack? And the way that it is done, we make a schematic for each FME in the payload, and by using different color bands, we can then mark which ampule is to be cracked at which point. So for in this case, the green band here, or right here, shows you that the astronaut is supposed to crack this at the activation time scale, and the blue band here shows that you are, that is the deactivation time. So if you have two timelines, one to activate, one to deactivate, the astronaut will know which one to use. In this case, you see that there actually are two green bands, and what that means, those both are to be cracked at the same time. In some experiments, you may want to have both ampules cracked at the same time to activate the experiment. In this case, there actually was an experiment where that was the case, and that way astronauts will know that too. In this case, you will have type 2 mixed stick, which is the long ampule, only one activation. And in this case, you have a type 3, but one of them is a control, so it's not cracked at all. So a lot of work goes into making sure that the timeline is accurate, and sometimes, and the idea is to make sure that everything proceeds smoothly, and usually that is the case. Well, finally, one thing that is important for all students to do after the experiments have been selected for flight and before they actually fly is to make sure that the experiment works exactly as you want it to do before it is flown. And as those of you who have flown experiments know, oftentimes you end up making some adjustments to make sure you get most out of experiments. I don't think there's been any experiment that has flown to date where it's flown exactly as originally proposed. There have been some changes. And what is important to remember that there are some things that you can change between experiment selection and actual flight, and some things that you cannot. And anything that has to do with the NASA Flight Safety Review, you cannot change anymore. So you cannot not introduce any new samples to your experiment. You cannot increase the amounts and concentrations of anything. So that is not possible after the samples list has been submitted to NASA Flight Safety Review. But you can decrease samples, including removing them. You can change special handling requirements, and you can even slightly modify the experiment timeline aboard the International Space Station with some constraints. Again, because by this time, we're already working with the ISS scheduling crew office to make sure that the timeline makes sense for the astronauts. So there are huge modifications, probably are difficult to make, but there's always been adjustments that can be made and have been made with Mission 1, and I'm sure there are going to be some for Mission 2 as well. And at the end of this process, just before flight, a few weeks before flight, we come up with this document, which I show the cover for here. For the last time we did that in February 2012 for Mission 1, where you basically confirm that these are your final details for experiment. That is really the point where you have to make sure that everything you want to fly is listed exactly as you plan to fly. The special handling requirements, for example, if you require refrigeration during transportation, is exactly as you want it to be, and the timeline aboard the station is what you want it to be at that point. After that point, it's going to be very difficult to make any changes. And that's why we want to do this confirmation of experiment details as late as possible, just before flight. So once we have confirmed that, we can then actually go to the point of preparing the payload. So once we have confirmed that you are happy with the way you have designed the experiments, you're done with the experiment design, final details, you will then load the experiments into the mini lab, the FME. Some of you have done that already. Those who are flying on Mission 2 will do that at the end of the summer. And then send it to NanoRacks for payload integration. And typically that's done about 4 weeks before launch. At that point, once NanoRacks receives the individual mix sticks from the different teams, They will be stored according to the special handling requirements. So if you require refrigeration during transportation, the mixed sticks are stored in the fridge. At some time before launch, at the specified time by nanoracks, just before they need to turn over the payload to NASA for payload integration into the launch vehicle, the nanoracks will prepare the mini labs for flight by drilling a pin through the open end of the Teflon tube, the one where you inserted the plastic end cap, drill it through, then heat seal level 2 and 3 containment bags so that you have sufficient levels of containment so that the astronauts are not exposed to any hazardous materials you may be flying. And then they are placed inside this payload box, which is designed to hold up to 24 mixed sticks or FMEs. They are placed inside here and then put into the actual payload handling device, which is shown here. This is a picture provided by SpaceX for the payload as it was prepared for Mission 1, where the whole payload was refrigerated by the use of this refrigerated container with cold bricks that kept the temperature at a very nice and cool of 4 degrees Celsius from the moment the payload was put together to the point where it arrived at the station. All right. So once the payload is put together, it's obviously put into the ferry vehicle, and for SSEP Mission 1, that was the SpaceX Dragon. And here are a few pictures of the Dragon capsule being prepared, including the cargo area where our payload was stored. And typically, the payload is moved into the launch vehicle about 10 days, a week and a half before launch. Of course, as those of you who took part in Mission 1 know, with delays, launch delays, it may get to be a little bit longer than the week and a half. And in the case of the first flight, it was about 4 weeks before launch that the SSEP payload was put inside the launch vehicle. And of course, once we are at this point, we are all ready to go. So finally, we can launch our payload to the ISS. So here are just a few images of the first mission, Mission 1 to the ISS that we did. So this is a picture of the launch of the SpaceX Dragon vehicle from Cape Canaveral, the grappling of the vehicle once it arrived at the ISS, and it was berthed into the ISS, the cargo being transported from Dragon to the ISS, and this is an image of the mixed sticks actually being handled right here. Not from this particular mission, this is actually from a mission that was done about a year ago to make sure that the mixed sticks work as planned aboard the ISS. But this basically shows you how the astronauts handle them. In this case, this is ISS Commander Kelly shaking the mixed sticks, which a lot of you have asked to do. So that really is the way we navigate the experiments from the moment you send in the proposal to the point where they fly and are being conducted on aboard the ISS. So as all of you know, finishing the proposal is not the end of the process. It's actually really just the beginning of a lot of work. But I've been delighted to hear that a lot of you really think it's worth all the work to fly your experiments in space. So I'm very delighted to hear that hard work has been worth it. And I'm looking forward to more talks tomorrow as well as the poster session today.